Good morning, everyone. Just a little bit of bookkeeping. We're going to push everything back 15 minutes, so this talk is going to end at 11:30, just so you know. So it's my pleasure to introduce Alex Smith. He is an assistant professor of molecular ecology at the University of Guelph in the Department of Integrative Biology and the Biodiversity Institute of Ontario. His research involves the discrimination of cryptic species and fragmented spaces using ants, parasitoids, and amphibians. He was also a Fine Outreach for Science Fellow in 2008. Here's Chris.
Thanks very much. I just wanted to start with a thank you. As you mentioned, I've been a Fine Fellow, or I had the privilege of being a Fine Fellow in Estes Park in Colorado in 2008 in May. I ran into the GigaPan and was kind of gobsmacked with it the first time earlier that year in March in 2008. There were some Google people with us down in some Googleites down in Costa Rica where I do a lot of my work, and we were looking at a bunch of different things, but in and amongst them, one of the programmers, Von Tan, had one of the beta units. And so I was kind of following Von around as he took photographs of us or of the Googleites looking at the ecologists doing what we were doing there. And I thought, I had this kind of Christmas lust. I was like, I want that. I really want to know more about that. And so luckily enough, when we got back, Vaughn had sent something to create. And so when the email invitations went out to come to Estes Park, to that fellow, the Institute got one of the invitations. And so I kind of kneecapped everyone else who was in a queue to get that invitation. I thought, I'm at the front of this line, I'd like to go. And so it's just with a real sense of gratitude and excitement that practically everything that I've had to do with this initiative since then has been one of the more exciting parts and unintended parts of my research program over the last two and a bit years. And so in today's presentation and in the project that's described in the paper associated with this talk, there's a real sense, for me anyhow, of I'm kind of running along in the middle of the parade with this. This is not something that I thought, hey, I'm going to form a parade and follow me. This is what I intend to do. This is the hypothesis I'm going to test, and here I'm going to set out to do it. This is much more of a, particularly with this project, something that I was challenged to do by a colleague who had heard about how I had this technology and how I was using it. Now, what I had intended to do and what I wanted to do with it right away, the way that I knew I wanted to use this right away was in my work in Costa Rica, in Guanacaste province in northwestern Costa Rica, where I work on volcanoes at different elevations from sea level up to the cloud forest, looking at ant community change. So the change in the species composition that we naturally expect to see with changing elevation within a volcano through time and between volcanoes. And so one of the things that I've experienced in tropical rainforests since the very, very first time that I ever had the privilege in being in one was a real sense of being awed and overwhelmed, not knowing how to describe what I was seeing, not knowing the phone book or the email address book to check, to ask what it was that I was seeing. And so that's actually, unfortunately, as I've become better and better at dealing with specific small slices of the phonistic community there, with the ants as kind of the exemplar taxa, my address book for the botanical community has remained relatively slim. And that's connected to the kind of the slimness of my field notes. I mean, we teach our students, we've all been taught ourselves, that this is a moment in time, that you have an opportunity and a responsibility to record with as much detail as you can, because you can't get, if you have the opportunity to get back to that space, you can never get back to that time. So to me, this was a real way of augmenting kind of the Victorian, but kind of the illiterate Victorian, botanically illiterate Victorian field notes that I was making on the ground, saying, well, where had I collected these ants? Well, it was green, or it was mostly green, and the canopy was this high above me, or this close to my head. And this is an opportunity to use this as a resource to expand, to greatly expand the capacity with which I can ask questions of the broader intelligentsia, the botanical intelligentsia, to say, could you please tell me more about where it is that I'm working and what I'm finding there. We also have the opportunity to use this on a research program that we have called the BioBus, which is essentially an RV that we've taken and put some really cool photographs on the outside. And we send it across North America to state parks in the United States and across all the national parks that have road access in Canada. And we send undergraduate students and volunteers out to gather, essentially, particularly in Canada, where with the benefits of the Assistance of Parks Canada, we have permits that are as broad as if it's not a vertebrate, you can take it. And we're sampling this wide, diverse community to add to a growing library of DNA barcodes. So essentially a DNA-based species library for species identification. We photograph all the specimens, but now with the helps of these and the excitement and the enthusiasm of these volunteers, we have the opportunity of adding habitat context to everything. So that for every group trap or passive array trap that we have these gigapans that spread from some of the ones out in Newfoundland and in Cape Breton in eastern Canada aren't there, but literally from coast to coast and down to the Guelph and west into California, we hope next spring. And we use it in a lot of ways within our, you've seen the poster that Andy and company have set out in terms of how they're documenting their collection, and we're trying to learn from what they're doing in terms of streamlining our processes of photo documentation of the collection of invertebrates that we bring back and trying to figure out how we can take gigapans, flatten them, do a pre-arrayed stitch to kind of create these streamline the process and make a high throughput kind of pipeline, photographic pipeline for the specimens, the tens of thousands of specimens that we're bringing back. And then we live on a university campus. There's a lot of opportunity for us to build new buildings, and we're putting up one. That's the institute that I work in is kind of this little building. And the older, more historical structure right here is gone as of a week ago. So we have an ongoing kind of a weekly gigapan of the construction and the emergence of this new Center for Biodiversity Genomics that we hope will be kind of the global home and headquarters for this initiative to barcode all eukaryotes. So that's kind of cool. And then, like you saw, most of us are probably all running around doing as well, as when we have groups of people together, we gigapan them. And there's something really kind of cool and sociological about looking at motion, particularly little bits of human motion that are captured with still photographs and then stitched together. Talking about, so this is a meeting at INBIO in Costa Rica of different parataxonomists and Costa Rican scientists talking about their plans for bringing together the next set of specimens that they're going to send up to Guelph to barcode. So those are the things that I knew I wanted to use the gigapan for. And the project that I described in the paper submitted here, and what I'll spend the next kind of couple minutes telling you about is the parade that I'm within kind of banging a drum on, but I certainly don't feel that I'm at the forefront of. I have no baton, which is probably good for all of us, because that's just a bad image even to have thrown out at you at this point in the morning. But the dairy bush is an area on campus that we use within the biology department essentially as an outdoor laboratory. And we send our students from first to fourth year there to design and to learn about study design and to learn about the ecology of the area. And so there's a kind of a look at, well, yeah, me and the bush itself and some of the fauna that we catch within it. So a little bit of background about Guelph and about the dairy bush itself. And don't be afraid by the year. I'm not going to start that far back, other than saying that Guelph is essentially, in a Canadian context, is a very new place. It's only actually existed since 1827. And it didn't grow out of a smaller community like a hamlet that became a town, that became a small city, that became a large city, that became a metropolitan center. Guelph grew out of, essentially, so the red star there, it was well off the beaten track right up into the kind of the 20s, the 1820s, until this guy, who's a Scottish poet, and in the particular vein of Scotland, a Scottish capitalist poet, who was sitting with his buddies from the House of Commons in the UK, and there had been an opportunity to buy large tracts of land in Canada. And so he purchased one of these with the help of what was called at the point the Canada Company, with the understanding that he would build a town. There's the intersection of two small rivers. Hydro was going to be a way to build several mills. So Guelph kind of grew out of nothing to a small city fairly quickly and fairly recently. It's an agricultural center. It has one of the largest kind of long-term agricultural colleges that now is comprised within the University of Guelph on our campus, agricultural colleges in Canada. And it's a fantastic kind of even within a Canadian sense, and I realize particularly if there are any Europeans in the audience, when I'm talking about kind of long-term things in Canada, we know that we're talking about kind of just a couple of moments ago, really. But it's a long-term sense for a Canadian institution to have access to some of the data about the campus. There's not a lot that we've forgotten yet. But there are some key things, and the Dairy Bush actually brought me into some of them. So the Dairy Bush is an 8.5 hectare woodlot on the extreme western part of the campus. So it's an easy, from practically anyone's office, and from mine in particular, it's about an 8.5, 10-minute walk, depending on how much snow there is. So it's an easily accessible locality for most of the students on campus and certainly most of the classes that we're teaching. It's also an area, so you can see here, the gentleman on the right is a colleague, a friend of mine, Kevin McCann, who is teaching a fourth-year course in designing and ecological methods. We play hockey together. Kevin is a much better hockey player than I am, and so amongst talking about the amazing goals that he scored while I was on defense, we talked a couple of times about GigaPan and about some of the things that I had brought back from various trips. And he said, well, you know what's really cool? I mean, my fourth-year course is going out. We have a small snapshot of time in which to gather our field data. Really, it's only a couple of weeks. The onus of the course is, of course, on the design of the experiments and then carrying them out as quickly as you can. You should come out and take a GigaPan of the course, kind of learning about the bush for the first time. And I thought that was a pretty cool idea. So we did. Now, I enjoyed it a lot. And that, to me, became one of these things where I was talking to somebody earlier today about the fact that one of the common academic trajectories, you get better and better at something until you get a certain job, and then you get taken away farther and farther from that thing that you got better and better at and you enjoyed. And this, to me, was, I enjoyed this a lot. I thought, this is only a couple minutes from my office. I'm going to try and keep doing this. And I thought, I'm going to come out seasonally. I'll take 4 GigaPans out in this bush over the next year, just to look at the kind of the passage of time, and maybe, who knows, maybe even I'll come out next year, and so in the end we'll have 8 GigaPans. And then I talked to Doug Larson. Doug, some of you may know, he's an emeritus botany professor at the University of Guelph, and he's also a luthier and a guitar player and a musician and a happier man now that he's retired. But one of the things, because he spends more of his time making instruments and playing them, but his expertise while he was working at the University of Guelph was in documenting these cliff communities of extremely, like thousands of years old, white cedars that live on the Niagara Escarpment very near the University of Guelph campus. And so he dealt a lot with the Dairy Bush. He was kind of the first person when he arrived in the 70s that started taking students out to this particular woodlot to look at documenting it and to look at preserving it. He's an activist. His soul is the soul of an activist. And there's been a lot, like you saw the construction GigaPans earlier, the buildings that are associated with my institute. We have a lot of construction pressure in and around the university, the holdings of the university. It started as an agricultural college, and one of the things that's happened is that more and more and more of our original holdings are being lost and developed upon. And so his challenge to me was, well, don't go out 4 times, go out, keep going out, multiple times, go out every week. And so that was essentially what I walked by on the south end of the bush to get to the bush each week is an ongoing development project. And so that is a snapshot. Now, that's one of his classes. What he looked at, remember we talked about James Galton kind of arriving in the 70s and in the 1820s, taking out all the trees that surrounded the city. That's what we had presumed had happened. Now, one of the use of the bush in these courses, one of the things that we had completed was a dendrochronological study. So we'd taken core samples and counted tree rings. And what we found was just in a random selection of the maples that are in the bush, some of those trees are first growth. They hadn't been cut. So they germinated in the 1780s. So kind of Napoleon Bonaparte was just graduating from high school at that point. And we're kind of now taking GigaPans with them. And that's, again, in a context, there are many older trees, but that's a cool thing. We didn't know this, and the students didn't know this, so they can go physically and touch this amount of botanical, of growth history. So a really simple setup. I mean, one of the beta testing units with some georeference points that are planted in the soil that I have taken some pains and been not as entirely successful as I would have liked at having them not get moved through time. But returning to the same point, and it's a Wednesday, it oscillates between a Wednesday afternoon, lunchtime, morning, evening. But if you're going to come to the city of Guelph and you want to have a picnic, come on a Wednesday. I've been doing this for 63 weeks now, and it's rained on me once. So we've got now an array of these gigapans through time. We've got seasonal captures, and I think one of the things for me, in terms of an overall theme of this, is I'm running within the parade, trying to figure out what my hypothesis is that I'm testing. I think one of the hypotheses, or one of the conclusions, one of the things that I've learned out of this initiative is there were the things that I planned to do with it, the international and the continental kind of surveys that I planned to do with this. This incidental occupation has become one of the most enjoyable parts of my week. And so I suppose the challenge, or my challenge if you're not already doing it, is think about occasional ways that you can use this technology locally. Think about it in terms of outreach, and this is the one from Wednesday, just before I got on the plane to come here. And I think one of the neat things is, one of the reasons I've enjoyed it so much, I suppose, is because of missed details. Things that there's the bush that you know, but then there's the part of the world that you don't know that you don't know. So that's a reduvid assassin bug with the shadow of the bug captured as I was speaking to one of the local journalists from the campus. And I was going blah, 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 blah. I didn't even notice that it was there. This is a moment that's captured that I can survey, that my students can survey, and that we can use within these courses to figure out and to develop essentially a relationship in the bush. We can look at elements of phenology. That's Sanguinaria canadensis, the bloodroot. And we've looked at there's a specific plant that we've now seen the phenology, the growth of that individual plant through time, so that when the students get there, practically no students are around when this plant flowers. But we have the snapshots, we have the capacity to annotate and to lead them and to direct them through this bush through time. We've extended essentially the periodicity of the lab, of the laboratory environment. We've added a digital component to the lab for between first and fourth years that are out there working. Surprising elements, I suppose, to me, of distribution and ecology. Lots of non-native species that we're capturing through time there. From a floristic perspective, certainly there's lots and lots of garlic mustard, so we can look at garlic mustard kind of first-year growth, second-year growth, and the extent of that along the paths, the pathways as more and more people move into the bush. This is a snail called Sepia nemoralis, a European snail, and the bush is full of them. And so this is kind of a link that I'm trying to make with a kind of a citizen science project in the UK to look at the distribution of these snails globally across the world. And I think from their perspective, one of the bits that I'm trying to cast towards them is the use of the bush as essentially as a digital lab to go out and look for snails, because there are a lot of them all over the place. And it can occupy maybe larger than your director would like to think portions of your day searching through the leaf litter looking for mollusks, which is hard to explain as a myrmecologist. So we use it as I mentioned, with the junior class, particularly with their online episodes, complementing and augmenting the specimen analysis that they're doing in the lab. Those are some notes for me. I apologize for that. You saw them, but there were some notes for me from some impressions that have come to me over listening to the fantastic array of speakers that have been arrayed here. And I just wanted to say a long kind of parenthetic thank you certainly to the Fine Foundation for having the foresight and the continued involvement in this project to bring more and more people into the fold and to think about science and how we use imaging within science. And it's been my pleasure to listen to all of you throughout the weekend. And these are some of the most exciting meetings that I go to, kind of the interdisciplinary and the high level of, I don't know, the excitement potential that comes out of this is just, it's really cool. So thanks a lot. And I look forward to hearing more this afternoon about what essentially the elegant use of a time lapse with GigaPan, because this was essentially a kludge, and I'd love to know a more clever way to do it. So thanks for your attention.
We have time for a few questions.
If you don't mind a technical question, the issue of getting pretty much everything you can in focus when you're working at that scale. Do you want to talk about that a little bit?
Yeah, well, it's a huge issue. I mean, one of the things that in, what would you call it, GigaPan 100 that you think about, and I guess how we're initially all, at least how I was taught to think about panoramas, was standing on the top of something tall, looking away. So setting your focus and locking it in at plus or minus infinity. When you're in the forest, you're in an environment that has huge heterogeneity in the depth of field. And so the way that so what did I talk about? I talked about a botanical setting, and I preceded it by saying, I'm not a botanist. And I'll precede this again by saying, I'm also not a photographer. But the way that I've dealt with it is essentially letting each frame autofocus. And so I do get more depth because of that, and occasionally, particularly adjacent to tree trunks that I'll get an extremely focused portion of a white pine trunk, and I'll look at a caterpillar there, and perhaps the frame beneath it is out of focus or is in less in focus because what it's focused on there is the bush behind the white pine tree trunk. So I don't think that's not ideal. That's less than ideal. It's kind of an operational solution to this problem, and I mean, this is now an opportunity for more clever people. I mean, what would be cool? Z-stacked, to look at those 768 images, each one of them stacked in depth, and then tiled together.
Well, what's the time per pick when you do that?
The way I'm doing it right now, it's a little bit longer. What have I got it set up? The time I'm using the beta unit, so it's a 2.5, 3 seconds. I'm kind of standing in the bush for about an hour. It could be quicker. But my impetus to reduce the time that I'm doing it is countered by the fact that I really enjoy standing in the bush.
I was trying autofocus one time recently, and there were several times when the camera couldn't make up its mind, and I had about a 6 or 7 second time.
It depends on the camera a little bit.
So then I'm using a G10, one of the Canon G10s.
That's one place where getting a better camera makes a huge difference. When you shoot them with a small one, it's like every.
Well, I was using a Canon EOS, I forget the